Hello everyone, I am Varun GV. Today we are going to see the topic on ensuring the drug safety in personalized treatment approaches. And these are the topics we are going to cover in this presentation: introduction, definition, precursor of personalized treatment, methods, benefits, challenges, examples of personalized treatment, references. And we are coming part of the introduction. So now widely we are believing that monitoring or preventing the disease must be tailored or personalized to the individual unique, like biochemical, physiological, environmental exposure and behavioral profile. So there have been many issues associated with the broad acceptance of personalized medicines on the part of different healthcare sectors such as physicians, healthcare executives, insurance companies and ultimately patients. So these challenges are revolved around a need to prove that personalized medicine strategies, especially since many tailored or personalized therapies such as autologous chimeric antigen receptor T cell, that in simple way we are going to call as CAR-T, and cell transplant therapies for certain types of cancer. And like mutation specific medicines such as ivacaftor, this is we are using for treating the cystic fibrosis. So, in this history, the motivation of personalized medicine strategies have emerged in the last few decades. Coming to our next slide, this is a definition of the personalized medicine. When we come through our personalized medicine is also referred as a precision medicine, is a medical model that separates the people into different groups with metabolic decisions, practices, interventions, and products being tailored to an individual patient based on their predicted response or risk of evidence. So here we are going to see the a subject or a patient, then like they are having a one subject, we are having a different metabolic rate, and so based upon their metabolic rate and metabolism, then we are going to prescribe few drugs. Like for they are depending on the metabolic rate, then we are going to prescribe one adult, like we are full dose we are going to prescribe. But some, if they are having less metabolism rate in their body, so we are going to be prescribe low dose of the medicines. And like we are having a different sectors of subjects, and we are going to prescribe different types of drugs according to a metabolic rate of the subjects. So the precursors of the personalized medicine, we are coming to a history. See here, the Garrod is a in English physician. He studied a phenotypic manifestations including alkaptonuria, albinism, cystinuria and pentasuria. In these, he focused on alkaptonuria, led to some notoriety when he observed that some members of families exhibiting the alkaptonuria showed measurably outlying values for certain basic biochemical assays from the urine. So this lead him to conclude that alkaptonuria was due to a specific altered course of metabolism among affected individuals, which was subsequently proven correct. Then Fisher continues the phenotypic variation with discrete heritable factors that contribute to this variation by suggesting that many factors, such as genes, might contribute in a small way to a particular phenotype. So Fisher's example we are going to see in based on height. An individual who are inherited only one of 25 genetic variants are known to be increased height. So an individual who are going to be inherent only for 10 or 12 genetic variants we are going to be seen in the average height subjects. And when an individual we are going to see in 20 or 25 genetic variants are known to be in a shorter height. So as a result, much of contemporary focuses on the personalized medicine is rooted in the findings of genetic studies. It has been shown that individual personalized treatment is safe and effective. So whenever we are coming to see the individual patient, then we are going to see in all these categories or sectors. So firstly, we are going to access the healthcare and genetics and genomics, imaging and radiology, wireless monitoring, environmental exposures, and tissue biomarkers. So these are the methods that we are going to see. And firstly, in order to know that mutation is connected to a certain disease, researchers often do a study called a genome-wide association study. What you call genome-wide association study. So, genome-wide association study will look at one disease and then sequence the genome of many patients and with that particular disease to look for a shared mutations in the genome. Mutations that are determined to be related to disease by the genome-wide association study then we used to diagnose the disease in a future patients by looking at their genome sequence to find that same mutation. So the first genome-wide association study conducted in 2005 studied patient with age-related macular degeneration. So they have done the studies and they are conducted in 2005 and studied patient with age-related macular degenerations. And these are the benefits we are going to see. Firstly, like personalized treatment helps care providers better understand the many things including environment, lifestyles and heredity that play a major role in the patient's health, disease and condition. This information gives more accurately predict which treatments will be most effective, safe and prevent the illness in initial stage. Benefits are choosing the treatment for preventing the illness, improve the disease detection, and then predict the susceptibility to disease, prescribe the more effective drugs, and customize disease prevention studies, avoid the prescribing drugs with predictable negative side effects, and reduce the time, cost, and failure rate of the pharmaceutical clinical trials. So these are the challenges we are going to see. As personalized treatment is practiced more widely, a number of challenges are arises. The current approaches to intellectual property rights, reimbursement policies, patient privacy, data biases, and then confidentiality as well as regulatory oversight will have to be redefined and restructured to accommodate the changes of personalized treatment will bring to an healthcare. So, a survey is performed in the UK concluded that 63% of UK adults are not comfortable with personalized data being used by medical field because like one subject a patient they are having a different metabolic rate and like they are used to prescribe lower dose and higher dose and like other few doses and like they are not comfortable with some few drugs, so they are going to be seen and like they are concluded with the UK patients 63% of UK adults are not comfortable with that personalized data being used in a medical field. So example genetic data they are obtained from next generation sequencing requires computer intensive data processing prior to its analysis. These are the examples of personalized medicine. The human body deals with traditional pharmacotherapies such as drugs to treat the disease in two ways. Initially, the body must respond to a drug. The drug must distribute it throughout the body and during this process, the drug might be biotransformed or metabolized into useful components which would being to elicit the effects. Finally, any remaining drug or drug components are excreted. These processes are often heading of pharmacokinetics and collectively called as ADME, that is nothing but absorption, distribution, metabolism and elimination. So we are coming through our examples. This is the example one how they are going to be seen in the personalized medicine. Firstly, warfarin is the example here, and warfarin is widely used blood thinning medication that if not dosed properly could cause the potential life-threatening adverse drug reaction. So warfarin targets here a particular gene that is VK0RC1 and it is metabolized in the part by the gene CYP2C9. So, naturally occurring genetic variation in both the VK0RC1 and CYP2C9 genes leads to a variation in the pharmacodynamic and pharmacokinetic properties of warfarin across the individuals, ultimately creating the variation in individuals' responses to a warfarin. This is the example one. We are seeing a example as warfarin, and this is the example two. The drug is nothing but primaquine. Primaquine has been used to manage malaria with some success in the parts of world where malaria is endemic. So, however, the doctors are working in the past observed that some of the soldiers they are treated for malaria that were provided the drug became the jaundice, so and anemic, and ultimately exhibiting symptoms of acute hemolytic anemia. So it has later shown that individuals are exhibiting acute hemolytic anemia after primaquine drug administration. So this is the variance in G6PD gene. So these are the references I would have referred for this presentation. Thank you.